So, welcome back to this class. While continuing the story of insulin, so as I mentioned that there are four key participants who played a critical role in unraveling or connecting the dots between pancreas, insulin and diabetes and glucose. And those players were Frederick Banting, who talked about, Charles Best, John MacLeod and James Collip. We haven't talked about James Collip yet, and today we'll talk about him, how he landed. In the last class we talked about how the experiments were done where duct ligation served to atrophy of the acidic cells that produce the digestive secretions, leaving behind only the islets of Langerhans, and it was figured out that pancreatomy was the method of inducing diabetes. And thereafter, in the summer, after a lot of setbacks and failure, however, the team reported in the fall that they were keeping a severely diabetic dog alive with the injection of extract made from duct ligated pancreas and prepared following MacLeod's instruction in saline. Amazingly, this extract dramatically lowered the blood sugar level of the diabetic experimental dog. This was the critical seminal experiment in 1921, let's say 103 years back. But then started a very peculiar. So, today's class will start with the findings are represented and the tensions begin. Here begins the next set of drama called human drama and human emotions, which plays a critical role whenever you do a discovery. This particular aspect of insulin is not very well known, but let's explore what exactly happened once they made such a seminal discovery. On December 30th, 1921, MacLeod, Banting and Best presented their finding at the conference in American Physiological Society at Yale University. Now you are in US, right? At Connecticut, Yale. Banting, out of nervousness and inexperience, did a poor job delivering the paper, and the audience was highly critical of the findings presented by Banting. MacLeod, on the other hand, as a chair of the session, joined the discussion in an attempt to rescue Banting from the scathing commentary. So, it's a very peculiar situation. So, Banting, MacLeod and Best, they were presenting the work about this seminal discovery of when they pull the extract or the secretion from the duct ligated dogs and inject it into a dog in which they have already removed the pancreas, and they could make that dog survive. But then whenever you do any such experiments where you have very little control, there will be always people who will be skeptical. So, when they were presenting their work in the month of December 30th in 1921 in Yale University, in a session where session chair was none other than MacLeod, other scientists were extremely critical and they almost attacked Banting and Best about highlighting the drawbacks of the experiments. And this cannot be right, because mind it, at that time it was a huge issue, and MacLeod came to rescue of Banting, but there was a misunderstanding. The misunderstanding was that after this fiasco, Banting became convinced that MacLeod had stepped in to steal the credit from him and Best, and the relations between the two began to deteriorate. So, on the contrary, for Banting it was as if MacLeod entered into the fiasco in a pretext to save Banting. He felt that MacLeod is taking the whole credit in his name. These are some serious problems in scientific communities. Who will earn the credit? Because at that time, problem of that kind means you are the top of the world. And this literally left a very bad impact for Banting and McLeod. And this was the beginning of a deteriorating and breaking away relationship. Next, what happened? Purifying insulin and the first human test. Mind it, so far it was all on dogs. You are moving from dog to human. At the end of 1921, McLeod, now comes that name, Collip. Remember, I was telling you that this one name is still missing from that list of 4 people who are involved in this process. James Collip. So now this name is entering the end of 1921. And prior to that, this experiment has already happened. So now, with the entry of Collip, there will be even much more interesting things will happen. James Bertram Collip, a biochemist in the Department of Physiology at University of Toronto, to help Banting and Best with purifying this extract. So, you are only working with the extract. You do not know what all chemicals are there, right? And this is not the era of mass spectroscopy and all other high performance chromatographies and all. This was a era before that. Okay. Collip, another University of Toronto graduate, was on sabbatical from University of Alberta and supported by a fellowship, had returned to his alma mater. As the experimental pace quickened, Banting and Best needed large amount of their extract, and Collip set to work purifying the extract for clinical testing in human. So again, they say you have to be in the right place at the right time. It's all about the timing. The first clinical test on a human patient were conducted on severely diabetic 14-year-old boy. Although the injection of the extract failed to have resoundingly beneficial effect, the Toronto team continued to experiment. A short while later, Collip made a breakthrough in purifying the extract using alcohol in slightly over 90% concentration to precipitate out the active ingredient. And this active ingredient is insulin. This was Collip's way of, from a mixture. So, this what is coming out from the pancreatic duct is a mixture of A, B, C, D, I, likewise. From that mixture to extract out the I, the insulin. Now, at the same time, the personal tension was mounting among the 4 scientists as Banting became increasingly bitter towards McLeod and pitted himself and Best against Collip in the race to purify the extract. At the end of January, we are into 1922 now, Collip came to Banting and Best lab and informed the two that although he had discovered a method to produce pure extract, which is insulin, he would share it only with McLeod. It was only Best quick restraint that stopped Banting from attacking Collip. This is a very ugly situation, and this is the situation which the story of insulin has gone through. And as we will proceed further, we will see there are many uglier side of insulin story. Fortunately, for the future of insulin, an uneasy agreement was made days later, allowed them to continue to work together. On May the 3rd, 1922, McLeod, representing the group, announced to the international medical community at a meeting of the Association of American Physicians that they had discovered insulin, the anti-diabetic agent, 1922, and they won the Nobel Prize the very next year. So think of the gravity of this discovery that led them to win a Nobel Prize. Banting and McLeod received the 1923 Nobel Prize in Physiology or Medicine for the discovery of insulin. That Nobel Committee chose only Banting and McLeod for the award, caused more animosity. Banting, outraged that McLeod was chosen to share the prize with him, immediately announced that he would split his winnings with Best, the graduate student at that time. McLeod, perhaps in reaction to Banting's gesture now, that he too would split his award with Collip. Those are the 4 names, mind it. Two won a Nobel Prize, other two got a share of the Nobel Prize, but they didn't get the Nobel Prize. Those who did not get the Nobel Prize was Collip and Best, while as MacLeod and Banting won the Nobel Prize. By the end of 1923, had been a commercial production for a year at Eli Lilly and Company laboratories in Indianapolis. Diabetic patients who received insulin shot recovered from comas, resumed eating carbohydrate in moderation, and realized that they had been given a new lease of life. This was the story of the Nobel Prize, and if you follow, some people were forgotten, and one of them was forgotten in this process was none other than J.G.R. MacLeod. 100 years ago, he was an Aberdeen scholar, was given one of the highest honors. The hormone produced by the pancreas keeps the blood glucose level under control, and Professor MacLeod has been described as an unsung medical hero that history forgot. This is fairly sad. We all know about the controversies and all, but we hardly figured out that what was his contribution at that time. 1876, and moved to Aberdeen at the age of 7. In the Granite City, he was educated at Aberdeen Grammar School and then studied medicine at University of Aberdeen. He went on to become a lecturer at University of Toronto, while in Canada, he teamed up with young surgeon Frederick Banting. So the initial work, glycosuria and all those things, were all work of MacLeod. So MacLeod could pull the connections between the pancreas, diabetes, glucose, and insulin. So there are 4 players. Okay? Pancreas, insulin, glucose, diabetes, urine, and others. So it was MacLeod who could put all of them in one go. What are the connections between all of them? And that's what set the stage for what we today know as the insulin therapy. So Banting isolated the internal secretion of the pancreas and succeeded in reducing blood sugar level. Working together, and after trials, two men published their finding in 1922. The breakthrough ultimately eased and prolonged lives of diabetes. The following year, the 2 men shared the Nobel Prize. So this is the picture what you see of John James Reichard MacLeod. Here you see Frederick Banting and Charles Best, and these were the dogs on the roof of University of Toronto. Frederick Banting and Charles Best. Medical Building, 1922. Dogs were used as experimental subject in insulin research. These were some of those extraordinary people, and the story of insulin is the story of these extraordinary humans who, amidst good luck, bad luck, successful experiment, unsuccessful experiment, criticisms from fellow colleagues, controversies, their personal vendettas and their personal egos, their mutual disagreement, yet science moved on. And humanity will always remember some of those classic experiments, which was this experiment, this seminal experiment, and then translating this after the summer of many setbacks and failure. However, the team reported in the fall that they were keeping a severely diabetic dog alive with injection of an extract made from duct-ligated pancreas and prepared following MacLeod's instruction in saline. Amazingly, this extract dramatically lowered the blood sugar level of diabetic experimental dogs. This is one seminal experiment, followed by another experiment which was done out here by James Bertram Collip. The experiment was purifying the extract, specifically this one. At the end of January, Collip came to Banting and Best's lab and informed the two that although he had discovered a method to produce pure extract, So, this pure extract discovery was one of the critical part of it, where Collip made a breakthrough in purifying the extract using alcohol in slightly over 90% concentration to precipitate out the active ingredient, the insulin. This was another crack of a discovery which completely changed the course of our understanding about how to extract insulin, how to extract several other such compounds, and how such extracts could be purified and you get the active ingredient and you use it to re-inject in order to test its therapy. But that brings us to a point that the next goal, obviously, how pharmaceutical industries develop, how we can synthesize these out of the body. And those are some of the biggest things which happen later. So, either you synthesize them organically or you synthesize them genetically using genetic engineering technique. At that point, there was DNA was not discovered. We are talking about a time when genes, DNA, these were slowly people were trying to understand. DNA structure was not known. It was in 1950s that Watson and Crick came with a model based on some of the crystallographic images. So, if we look at the story of last 103 years of insulin, we'll realize how different other discoveries, say structural chemistry or structural biology, how it shaped up, how we came to know about, in the next class what we'll be doing, about the structure of insulin, how we do all the binding assays for insulin, how these binding assays helps us to develop different kind of sensors for insulin, what are the role of radioimmunoassay and ELISA, enzyme-linked immunoabsorbent assays, for quantification of insulin, followed by how insulin could be produced by organic route of, when we talk about organic route, it is basically a peptide hormone. So, a small peptide hormone, how could you synthesize it using synthetic peptide chemistry, followed by the discovery of using genetic engineering tools for developing methods of producing them in large amount, and how today's world still has lot of issues with the production of insulin and how it is influencing the life of human being who suffers from diabetes. So, there are lot of challenges which are coming up, and more diabetes is becoming prone in the human society. We are exploring newer and newer ways to cut down on the prices of insulin production. And in the next class, we will talk about the chemistry of insulin and how it is shaping some of the future research areas. Okay. So, with this, I will close in this class and we will move on to the next class. Thank you.